Hello everybody. I'm going to cover the viruses here as a substitute for our Monday lecture since I won't be there. And yes, I'm recording at home, so if any dogs bark, those are my two dachshunds, Bert and Ernie. And if you see any kids walking around in the background, you know they got tired of hiding in their rooms while I'm recording lectures. So Chapter 5 in your textbook deals with viruses, and we're going to hit some highlights from that chapter. And I've got a little info that I'm adding in of my own toward the end. I'll probably chop this up into probably like 3 or maybe even 4 separate little video clips so you don't have to watch it all at one time if you don't want to. Alright, so some of this we've talked about before. Viruses are obligate intracellular parasites. We've talked about that over and over. We've also talked about how they're acellular. That means they're not composed of cells. They go inside cells to multiply, but a virus itself is not a cell. And in a nutshell, what a virus consists of, now there's a lot of variety out there in terms of the specifics, but in general, a virus particle itself consists of the genome. That's the genetic material of the virus, of course. And here we have a generic virus, and that squiggly little purple thing there is supposed to be the genome of the virus. Notice it's labeled as nucleic acid, and that's because viruses can have, depending on the type of virus, some viruses have DNA genomes and some viruses have RNA genomes. Alright, and then the genome has to be protected because when a virus is traveling from host cell to host cell, or it's out in the environment lurking around waiting for a host to come and pick it up, it has to be protected. You've got to maintain the integrity of those genetic recipes there on the genome. So the genome is going to be protected by a capsid. Capsids are built from proteins. They are viral proteins. That means the genes for those proteins are actually located on the virus's genome. And then outward from there, this generic virus we're looking at here doesn't have any additional layers, but as we'll see, other viruses will have, some viruses will have additional layering outside of this capsid area as well. And lots of, as far as virus morphology, what do viruses look like? What is their shape and their structure? This image here just gives you kind of an overview of some of the variety out there. So some viruses kind of look like this. They have this geometric configuration that's called a polyhedron, that type of geometric figure, and many of them do look like that. Some of them will have spike proteins that stick out from the surface. Those are usually involved in helping the virus attach to its host cell. You can see over here HIV is more or less spherical in shape, and it does have spikes protruding from its surface as well. Rabies looks like a bullet, believe it or not. Influenza is kind of like HIV, looks more or less spherical in shape. And this weird guy over here is called a bacteriophage. That is a virus that infects, uses a bacterial cell as its host. And some of those look strangely alien. So they have a capsid head up here that kind of looks like this one over here, but then they have these other parts that make them look like, almost look like alien creatures, and those are entirely built from proteins. And these things look like spider legs over here actually help those viruses dock onto their host cells, and this little cylinder you see there is actually used to help inject the genome that's up here in the capsid of the virus into the host cell after that virus docks onto the surface. So there's a lot of variety out there with viruses, as we'll see. Okay, so just like we've used this word morphology before to refer to shape and structure and so forth, viruses have morphology as well. And more specifically, you can think about just the capsid morphology, the capsid of the virus, which is this protein coat that protects the genome. It can have a polyhedron type shape like you see here. In other viruses, the shape is helical, so spiral in configuration. Like you see over here with this virus, that little pink line right there is the genome of the virus, the nucleic acid, and all those little things look like purple peanuts are capsid proteins, and they are arranged in a twisty spiral configuration around the genome. So if your capsid looks like that, it's called a helical capsid. Then some viruses are like this one over here. They're called naked viruses. They don't have any additional layers, or they can be called non-enveloped viruses. Many viruses, though, have what we call an envelope. And what an envelope is, it's a phospholipid bilayer, which means it's basically a part of the host cell's membrane. These viruses steal a portion of the host cell membrane and they wrap that around the capsid usually as they're emerging from the host cell. There's some variety with that also. So this virus that we see here has a helical capsid on the inside, and then this blue and yellow outer layer that you see there is called the envelope or envelope, and that's a phospholipid bilayer. It's a part of the host cell's membrane that it stole. Now this one also has another layer. You see there in green, there's another protein layer there that's called the matrix, and some viruses have that and some don't. There's lots of variety. Envelope viruses will always have glycoproteins protruding from their surfaces. They kind of look like little spikes. Those are sugar protein combinations. They're proteins that have sugars attached to the amino acids in the protein. And, at least some of those are going to be involved in helping this virus attach to its host cell. And then you wind up with all kinds of different configurations. So, for instance, this virus over here has a polyhedral capsid that kind of looks like this. There's its genome on the inside, but it has an envelope as well. And if you notice, it has more than one kind of glycoprotein that are highlighted there in pink and purple and orange and so forth, protruding from the surface. So lots of variation with virus morphology. Lots of variation with sizes. Here's a size comparison from your textbook. They're showing us, here's a yeast cell like the Candida albicans yeast or the Saccharomyces yeast that are used for beer and wine production. They're about that big. 7 micrometers in diameter. Here's an E. coli, about 2 micrometers long. There's a Streptococcus bacterial cell that's pretty tiny, about 1 micrometer in length. The largest virus that's known is called Mimi virus, and not a human pathogen. But not all viruses infect humans. There are viruses that use anything you can imagine as a host. Probably it wouldn't surprise me if every living thing on the planet has at least one virus that can use it as a host. So Mimi viruses are pretty big. That's 450 nanometers, which is 0.45 micrometers in diameter. So that's actually a little bit wider than these guys. These are called Rickettsia bacteria. So they're bacteria and they're only 0.3 micrometers in length. Rickettsia, some of those use ticks as a host, and they can be transmitted to us by ticks, and there are rickettsial diseases that humans can pick up from tick bites because of that. So that's big for a virus. More typical virus sizes, you see some variety over here. Herpes simplex viruses, 150 nanometers. A nanometer is a billionth of a meter. That's pretty big for a virus. Rabies, 125 nanometers. There's HIV, 110. Almost the same size as influenza, 100 nanometers. And on down the line. There are some little bitty ones as well. Polio virus is only 30 nanometers in diameter. The yellow fever virus, a famous virus from the past here in the southern United States, about 22 nanometers in diameter. And that's not much bigger than some proteins, like the hemoglobin proteins, which transport oxygen around in your red blood cells. They're only 15 nanometers in diameter. So some viruses are so small, they're not much bigger than some of the proteins that are used in our bodies. All right, how are viruses classified? Now today, the genome of a viruses typically have much smaller genomes than any type of cellular living thing. So we have the genome sequenced for probably just about every virus that has been identified, and certainly almost every human pathogen uh, has had its genome sequenced by this point. So a lot of the classification today is based on genetic relatedness. You look at the sequence of the genome, and that AGCT or AGCU letter code you have there, more closely related viruses are going to have more closely related matching patterns among their genomic sequences. But classically, how viruses have been classified, it's based on, okay, this genome on the inside, is it RNA or DNA? Do they have envelopes? Yes or no? Some viruses just have this capsid shell and that's it. Some have this phospholipid bilayer, and some are going to have some additional layering in between, like that green matrix we were talking about a few minutes ago. What is the shape of the capsid? Capsid shape has been used to help classify viruses. Strategies the virus uses for copying its genome, we'll take a look at some examples of those shortly. And usually these things like this, when you make comparisons, if you have two viruses that are very similar along all of these lines, when you look at their genetic relatedness, their actual genome sequences, they're going to be pretty similar. So the old school methods for classifying viruses have held up pretty well, even now while we have all this great genetic information out there to help us make comparisons. All right, the genomes. I mentioned that the genomes of viruses can be either DNA or RNA. So different types of viruses have differences in that regard. And so this is a table from your textbook, just pointing out some examples. You don't need to memorize these. We're just pointing out that there are some different types of viral genomes. So among your DNA viruses, those are viruses that have DNA genomes inside the virus particle itself. Some of them have double-stranded DNA genomes, just like our cells have double-stranded DNA, or the cells of any living thing have double-stranded DNA as their genomes. But then there are some that have single-stranded DNA genomes. So there's something that's different from living things. So the smallpox virus, also known as variola, has a double-stranded DNA genome. The herpes viruses are double-stranded DNA, and so are many, many, many others. The parvoviruses are an interesting type of virus. They have a single-stranded DNA genome. And a parvovirus is there's a canine parvovirus. It's a very severe pathogen in dogs, and then there are human parvoviruses that tend to cause diseases that are not quite so severe, such as erythema infectiosum causes a reddish rash, especially in kids. Better known as Fifth's disease. Or I think sometimes it's called the slapping disease as well, because the kids look like they've been slapped when they become infected with that virus. Among your RNA viruses, we have some variety as well. Many of them have single-stranded RNA genomes. Some have double-stranded RNA, and then there's a special case where the genome inside the virus is an RNA molecule, but when the virus infects its host cell, it actually converts that RNA into DNA. So they do that through a process called reverse transcription, because they're doing the backwards or they're doing the reverse of transcription. Transcription, you make an RNA copy of DNA. Reverse transcription, you're taking RNA and making a DNA type copy. And then notice on here, among our single-stranded RNA virus examples, some of them are called positive polarity RNA viruses, and some are called negative polarity RNA viruses. And that doesn't have anything to do with electrical charge, so don't get confused by that. That is actually referring to if the genome in here, it's going to get released into the host cell. If the host cell then sees that as a messenger RNA, so that your host cell thinks, oh, there's the messenger RNA, ribosomes hop on and they start making the virus's proteins. That RNA genome is said to be positive sense or positive polarity, or it's a plus strand RNA genome. There are several different designations for that. Then other viruses have negative polarity RNA genomes, or they're called negative strand RNA viral genomes, or negative sense RNA viral genomes. And what that means is, okay, so that RNA genome gets squirted or released into the host cell, and the host cell doesn't immediately see that as a messenger RNA. Hang on. You've actually got to make a complementary copy of that RNA. You've got to, and that complementary copy will be a messenger RNA that can be translated to make viral proteins. We'll be talking more about that shortly, though. All right, so I think I'll cut off the first little video segment here on viruses, and we'll pick up here. We're going to start talking about viral life cycles and what viruses have to achieve in order to reproduce once they enter into a host cell.